In Lesson 3 of Genetics and Biotechnology, we're going to look at how DNA was discovered. So you'll watch this video, you'll open your Genetics and Biotechnology manual here, and you will read Lesson 3. If you're reading Lesson 3, it starts on page 24, so you can scroll down to page 24. And then you'll visit the Progress of Science timeline. That's linked here. So we'll click on that timeline. We'll be using this timeline. On the welcome screen, you can just click close, and I will show you how to use the timeline in just a moment. So keep a tab open with your timeline. And it also says you'll fill in the timeline research document with Cami. So I'm going to go ahead and open that. Okay, this will be something you'll be using while you do research of the timeline. So let's go for a minute to our lesson and explain what all this lesson is about. It's called How Was DNA Discovered? And again, we're in our story of the Miller family. Mary has gone to her physician. She's done a family health history. She has had DNA testing done. And while she's waiting for her results, Mary uses her computer to find out more information about hemochromatosis. And today you'll use a digital timeline for you to learn more about the dawn of molecular biology. So who are Watson and Crick? In 1953, James Watson and Francis Crick published a structure for the DNA molecule. This beautiful model explained the previously confusing data and fit experimental results so well that it was replicated and accepted by the scientific community very quickly. So these two guys are pretty much given the credit for the DNA model. Here's one of their early sketches of the DNA model. Here's actually a photograph of their model, and it actually is very huge. In this picture, it's hard to see the scale of it, but the scale of their huge model is just outstanding when you see it, like from a video of it or something like that. It's hard to see the scale of it in this picture. But their model had several features that had been missing from earlier proposed structures of DNA. The Watson and Crick model featured phosphate groups on the outside of the molecule and was made up of two chains of nucleotides that ran in opposite directions, held together by hydrogen bonds between the bases. And later in this module, we'll be actually building a DNA model out of connects, and you'll better understand those phosphate groups and nucleotides and hydrogen bonds when we build our model. So we'll get more into that later. Today, though, we're going to focus on this timeline. In 1962, the Nobel Prize in Physiology or Medicine was awarded jointly to Francis Harry Compton Crick, James Dewey Watson, and their other colleague, Maurice Hugh Frederick Wilkins. There also was another person who was in their research group and who helped them a lot. Her name was Rosalind Franklin, but she sadly died before they actually got the model built. And the Nobel Prize is not given to people posthumously, so she did not actually receive credit in the Nobel Prize award. But they received the award for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. That's actually what was stated when they won the Nobel Prize. So, what discoveries, inventions, or innovations were the necessary building blocks for Watson and Crick to discover the structure of DNA in 1953? They didn't just find out that information from nowhere. There actually had been lots and lots of work done by scientists throughout the years that actually led up to the discovery and the structure of DNA. So we're going to use this timeline. This timeline is found at timeline.hudsonalpha.org, but I already have a link for that to you or for you in the assignment. And so when we use that timeline, we're going to fill in some missing information on our chart. It will have years, it may have the who, it may have the what, and it may also help you out with why this event was important in the foundation for the work of Watson and Crick. So whatever's missing on this document is what we're going to do research to find. So the first one, 1665, Robert Hooke discovered cells using an early microscope. That's kind of our starting point when we get to the timeline. Now this section right here is things for you to think about. They're not going to be directly on the page for you to read, but you will have to use your knowledge of what you have found out about DNA and tell me why you think this event was important. Like, for example, why was the discovery of cells using an early microscope important to the eventual work of Watson and Crick? Well, this was the actual discovery of cells, and without the discovery of cells, we would have never have discovered what was in cells like DNA. So the discovery of cells was one of the very earliest and most pivotal parts of this information cycle. So on this part, you're going to have to use those brains and think. The answer's not just going to pop off the page and be there for you. You've got to use that noggin and think a little bit on this part. Okay, so this ends up having 2 pages. It ends on the 1952 with Linus Pauling, but then it tells you to select a biotechnology event from the last 10 years. So you would scroll up to the years 2010 to 2020 on the timeline and pick an event from that time period that you think would be worthy enough to put on our chart. Some discovery within the last 10 years that has built on the work of Watson and Crick. And then in summary, here are a few questions for us to think about. Why do you think the invention of the microscope was a critical event? Why was the development of X-ray crystallography a critical event? Why are Linus Pauling and Phoebus Levine included on this worksheet when their proposed structures were ultimately proved to be incorrect? Okay, so let's escape from this and go out and look at the actual assignment. Okay, you have your timeline that you'll fill out using Cami, and you have the Hudson Alpha timeline. So let's think about where we start here, 1665 with Robert Hooke. So I'm going to now go to the Hudson Alpha timeline, and I'm going to actually enter the timeline and scroll. Let's see. Sorry, I'm having a little technical difficulty here. I'm going to pause for a second so I can get the website to work. I'll be right back. Sorry, I was having little technical difficulties there, but here we go. If you are on the timeline, you can use your arrows, your arrow keys to scroll across the timeline. And remember, we're looking for 1665, so we keep scrolling until we find 1665. There's 1660. Here it is, 1665. So if I click on the 1665 entry, it tells me about Robert Hooke's discoveries using the microscope. And so when I go back to my document, 1665, Robert Hooke discovered cells using the early microscope. Why was this event important? Foundation for the work of Watson and Crick. Well, here I might put something like, without the discovery of cells, we would have never discovered DNA. Or if you would like to put some other comment there that applies, then that would be fine. Now, our next stop on the timeline is 1802 with Franz Bauer. So I will go to my timeline, close the part about Robert Hooke, and continue to scroll forward until I get to the year with Franz Bauer. Okay, I'm still scrolling. Here we go, and now we're in the 1700s, 1795, 1802. Here we go, 1802. The cell nucleus is discovered and first described by Franz Bauer. So when I click on that, it tells me that he discovered the nucleus. And why is this important? This is important because DNA is stored in the nucleus of the cell. And so I would go back to my sheet, I would tell what he discovered, why was this important, and then I would move on to 1865. 1865, we're going to find out who was the father of genetics. I'm going to do this part with you, and then I will let you continue the rest on your own instead of including it in the video. Okay, so we're looking for 1865. Okay. 1861, 1864, here we go, 1865. You'll notice we have two different people, but the person we're looking for is the father of genetics. He is not known as the father of genetics, but here's a cool fact. Remember David and Mary and their family, the Miller family, hemochromatosis? It mentions hemochromatosis. It actually was first discovered in 1865 by Armand Trousseau, but the term hemochromatosis was not given to the disease until 1889. But he's not the father of genetics, so I don't need him for this part of my timeline. But I do have another person here, Gregor Mendel. Gregor Mendel, we know from our earlier studies of him a few weeks ago that he is actually known as the father of genetics. And so here's who I'm looking for. Mendel's laws of genetics. He's now considered to be the founder of modern genetics. Through his experiments, he came up with his law of segregation, law of independent assortment, law of dominance. And so we can tell what he did, who he was, what he did, and why was this important. Then you will just continue on 1870, 1910, 1934. You'll just keep scrolling along your timeline and finding out the rest of the information for your graphic organizer of the Foundations of Discovery. When you're through, you will turn in your assignment, and then the next time we will continue on with our Miller family in Lesson 4.